Good morning, everyone. They asked me what song I wanted to play when I came up, so I picked a very Boston song. I heard backstage some people ended up in Hong Kong last night, so that's a real Boston thing to do, so I appreciate it. Really seeing the cultural heights of Boston. All right. So yeah, I'm really excited to be here. I'm glad everyone showed up early on a Friday morning. This is great. And I just want to give kind of a broad overview of some perspectives that I have on in general, the application of genomics today and what it is that we're doing. And then I'll touch on some of the, I think, hopefully transformative projects that we're working on together with PacBio and our scientific and clinical collaborators. So at a high level, most people locally will know the Broad Institute. And to sum up sort of at a very high level, our mission, it's sort of to understand the underlying causes of disease and close the gap between biological insights and impact for patients. And I think more than ever, it's really important to keep that in mind, this idea of connecting our science to society and show the practical application of what we're doing and why it is important and why it makes meaningful changes in people's lives. And I see if you watched the news yesterday, you'll see there's a real feel-good news story about sort of a gene therapy that has hopefully had a big impact on some newborns' lives as well. So again, not to oversimplify, but what does it mean? What does that actually mean? What are we doing at an institutional level? And then I'll drill down a little bit more into what we're doing. So you could kind of bin in one way of looking at it is that we're facilitating people, scientific researchers at the Broad and around the world in making discoveries, developing diagnostic markers, and ultimately moving towards new therapies. And so if we go down one more level, what does that actually mean? So what do you need to do to make discoveries? There's lots of things you could say fall into the bin of data you need, of activities you need to do discoveries. But one way of thinking about it is what are the sort of things that you need, or what are the attributes of activities and data sets that you create to enable discoveries. And what I'm kind of phrasing it here is one idea is populations. So you need data on a lot of people, as many as you can get, both healthy and with some known disease. So if you can create that sort of large-scale data set and have as many different data types as you can from that data set, genomic, proteomic, metabolomic, epigenomic, have associated medical records and understand phenotypes and family history, exposures, all of that. If you can bring all of that together, that's really where we're going to start to make these discoveries on the associations of genetics and disease. Moving into then the next phase of like diagnoses or diagnostic markers, or how do you create sort of modalities to identify what underlying condition a person might have. To power that, you sort of need to be able to capture phenotypes, describe symptoms. Then for any given patient, you need to be able to take a comprehensive view of the right data types to be able to make those diagnoses. And it will vary between whether you're doing cancer, rare disease, rare childhood disease. There's going to be nuance in there. But ideally, you want to capture some clinical phenotype, and then you want to put the best data in front of geneticists, pathologists, and folks who are going to make that diagnosis. And typically, we're not doing sort of population scale diagnoses. Now we're talking about sort of smaller numbers of actual patients as opposed to participants. And then moving that into, well, what do you do about that? How do you develop new therapies? Again, there's lots of ways to do it, but one clear way is to mine the data you've got up front to identify biomarkers, so indicators of the disease pathways, to do more on sort of describing the functional activities that happen, the functional genomics, to get to the pathways that are important in any given pathology, and then start to identify within that pathway what are the things we could target, whether that's with small molecules, biologics, gene editing. So that's, so together, this is sort of the span of things that we support at the Broad Institute and largely in sort of the genomics community, I would say. So rather than dive into all of these, I'm going to sort of focus a little bit on maybe what we would consider our flagship data type in genomics, which is the human genome. Of course, there's many other data types that we work on, from infectious disease and microbial and all kinds of types. But the genome is sort of where it started to some extent, right? The sort of the basic fundamental building block. And you could say, well, the genome's kind of boring now. It's easy to sequence genomes, right? And you kind of like, why do you need to sequence the genome? Why don't you really focus on a lot of like very bespoke, custom, high-tech, new molecular assays? But actually, the genome, I think, remains one of the most powerful things we can do. And in fact, sequencing the genome has impacts across our entire lifetimes. And so, there's a lot of really interesting things happening in science right now and in translation and in the clinic that all can or should or are using the human genome as a baseline or as a starting position. And so if you think about a sort of a timeline even of our lives, a new exciting area is this idea of somatic mosaicism. It's been, the concept's been around for a very long time, but now we're starting to see investment in research with folks certainly in the UK. Sanger Institute has done a lot of work on this. And now in the US, there is a national consortium funded by NIH to really understand baseline somatic mosaicism. Now, if you're not really in that field, what does that exactly mean? Well, we were always taught that essentially, your genome, the DNA encodes the blueprint of your body, and it's the same in every cell of your body, and that's the thing that's sort of static and fixed, and then other things can happen over time. In reality, we now know that actually from the earliest stages of embryonic development, once you start to differentiate cells, it's like, ectodermal layer, endodermal layers, you actually start to get individual changes, small, very, very small numbers of changes in the DNA code in different cells. And so these are somatic because they're not encoded. They're not sort of the thing that got passed down from the parents. And this continues as you continue to differentiate and as tissues start to form. And so actually, even though our sort of standard way of thinking about it is that every cell in our body has the same DNA, realistically, there's actually differences. And it's sort of tissue specific. And as you get older and older, these differences start to accumulate. And so there's a clear relationship between age and somatic burden, or the amount of sort of non-germline inherited somatic changes in your DNA code that differ across different cell types. So this is fascinating. And then you can start to measure that. And so now we have methods, of course, we've developed methods to start to catalog the rate of somatic mosaicism. And so mosaicism clearly just means, we're sort of this tiled mosaic of cells that have slight differences across our body. So we can measure somatic mosaicism now using lots of different methods. And so from the sort of for the technological perspective, it's not that dissimilar than like somatic variant discovery. So you're really looking for low-level somatic variants across tissues. And so one of the programs, the NIH program, is SMAHT, S-M-A-H-T, which is somatic mosaicism across human tissues. It's cool to say it in Boston because it sounds like SMAT. And then when you apply for grants, of course, you have to put a name on your particular center, and we're one of the genome centers for the SMAT network. And of course, which is a collaboration of sort of Broad and MGH and others, our center is called WICKED-SMAT. And we tortured the acronym to make WICKED work. It's something like whole genome knowledge. We really tortured the acronym, but we were like, we have to make it WICKED-SMAT. Anyway, SMAT is the network. Interesting enough, if you start talking about it, saying SMAT a lot gets really tiring after a while. But anyway, So it's a network. So it is a network now, like 5 genome centers, lots of different groups who are really working on how to profile different tissues with various technologies. So we could do short-read genomes. We're also doing long-read genomes. You can do bespoke custom assays to go really deep in certain areas. But and this is sort of recapitulating some work that's already been done at the Sanger and other places. But what's clearly emerging is this really interesting set of signatures of the types of somatic mosaicism that occur in different tissues. And there's things that you might sort of expect. So interestingly, like one of the biggest organs in the body is your skin. And skin shows a lot of somatic mosaicism, not surprisingly, because it sort of is the barrier to the outside world, and it gets a lot of UV damage and other things. You can see lots of somatic events that happen in the skin, but then other tissues and organs will have different levels as well based on their sort of different exposures. So that's a fascinating area. And of course, it's really about what's the baseline and how do you know, because the issue, of course, is like at what point does somatic mosaicism flip over into sort of early cancer or start to like flip into a pathology that's actually important to know. So that's like, something I hadn't thought about a lot, for forever until we started this project, but it is really fascinating. So then that's sort of like from earliest development. When babies are born, if you're born healthy, there's already tens to dozens of tests that are done on healthy newborns. So in the US, at least our state Department of Public Health mandated newborn screening programs that often use mass spec and other sort of low-cost assays to test for a small number of actionable conditions where it's important to know it as early as possible because you're going to do something. There's an intervention to take to avoid a bad outcome for a newborn. So many studies now have, pilots have kicked off to say, well, what if you sequence the genome of a newborn? What would you learn? And there's already reports that you could test for far more things. You always have to straddle the line of what you should test for in a nominally healthy newborn child. But there are many things that you actually might really want to know as soon as you possibly can because you want to do something about it. Of course, if you're not born healthy and you end up in the neonatal ICU unit, then it becomes a critical situation. And folks like Stephen Kingsmore and Rady's Children's Hospital and others have shown the value and sort of the urgent value of doing NICU diagnostics using whole genome sequencing, because you don't really have time, and in a newborn, you don't really have the ability to do a lot of like phenotypic measurements to really triage into like some specific gene test that you should do. So just a really quick, as fast as you can do a genome could be the way to do this, and it becomes sort of an implementation issue, and how do you do it quickly? As children grow older, you start to see the manifestation of other rare diseases, developmental delay, and other things that happen as you sort of either fail to hit milestones in development or start to exhibit other phenotypes. And this rare disease odyssey is something we'll talk about, but it is a big focus for more comprehensive data types like long reads, because you really want to be able to look for every possible type of variation in there. I've touched a little bit on the idea of populations and how important it is to have really well curated, rich data catalogs of large numbers of humans across populations so that we can properly understand the diversity of variation across different ancestral groups, so we can really catalog rare variants. And one of the things that really limits or can be a blocker for folks working in rare disease research or diagnostics is when you come up with variants and you have a variant of unknown function or variant of unknown significance. And part of that is just it hasn't been seen before. And so you can't say for sure this is a causative variant in this disease. And so this limits a lot of what can be delivered back or reported back or acted upon. And the way to really get to knocking down the numbers of variants of unknown significance is to try to see all the variants. And so, sequencing at the population scale is really, and then having systems in place to share that data, to aggregate the data, to train models on the data is really powerful in sort of understanding the actual impact of variation in disease states. And if you can do that, what you also can do is actually power sort of this nascent field of preventative medicine. And we talk a lot about sort of the burden of sort of the cost of the healthcare system and how much, certainly in the US, how much is spent on the healthcare system and sort of the poor outcomes. But very little of our comparatively, very little of that investment goes into preventative medicine. But what we've started to see as the population data sets with associated phenotypes and diseases are released, starting with things like UK Biobank and then leading into the US All of Us program, is that with enough people, you can create models of disease risk. And so looking at not just sort of monogenic single variant associations with risk of disease, but also building polygenic models, understanding the transferability of those polygenic models across different ancestral groups to identify folks that are at higher risk of developing chronic diseases, heart disease, diabetes, cancers like breast cancer, prostate cancer, colorectal cancer. There are markers in your genome that could put you at a higher risk of developing these diseases. The sooner you know that, the sooner you can actually take steps, whether that's with actual medical intervention, like going on statins, or more frequent screenings, or lifestyle changes. If you can actually implement that in a meaningful way and meaningfully reduce the number of folks who go on to develop those conditions, or at least ameliorate the impact of those conditions, you have a massive impact potentially on morbidity and mortality. And ultimately, that actually saves the healthcare system money. So I'm a big fan of this idea of sort of preventative medicine and disease risk, because these signals exist in your genome. They're there today. And if we know how to mine them, we could actually all be better informed. Cancer is an obvious case in which sort of genomically driven action, triaging people not by where their tumor shows up, but by what the molecular profile of the tumor actually is to inform the correct treatment regimen, and then going on to monitor after treatment with curative intent through MRD testing and other things. That genomics has had a massive impact on the cancer field, and of course, like I mentioned, in common disease as well, there's an opportunity for impact. So combined, all of these things now can be done from a simple whole genome sequencing assay, right? Not the most complicated molecular biology we could ever do, but just the basic whole genome sequence that we do. In our group at the Broad, we've done over 750,000 human genomes now. We did 100,000 last year. And so the data sets are just getting larger and larger and larger, and we really are at a great opportunity to mine these. And these are not all just sort of theoretical areas that we could be working in. These are areas that we and our group are working in actively today with partners around the world to forward the application of human genome sequencing to these areas. So I'm going to go from sort of the high level, what could all we be doing, and focus on maybe two use cases specifically. One thing is this idea of large-scale aggregated data sets to drive forward the discoveries, develop new diagnostic modalities, and ultimately get into therapies. The All of Us program, which most people will be familiar with as sort of the US version of the UK Biobank program. So it's the national sort of biobank program, which has enrolled to date over 800,000 US residents across the country. There's participants from every single state. Over 500,000 of those participants now have whole genome sequencing and microarray data. The vast majority of them also have connected medical record information. There's survey questions. So this is a pretty rich data set. And if you've been looking at, I mentioned polygenic risk scores, people have done a lot with the UK Biobank data set, because for a long time, that was sort of the only data set that was really widely available for researchers to access and to start to build models. But the UK Biobank data set is very specific to sort of the region in which it's been sort of recruited. And so if you develop models only from UK Biobank, if you develop a polygenic risk score from UK Biobank, it's going to be accurate for me, because I grew up in Ireland, which is not part of the UK, to be clear, but genetically fairly similar. And so it's going to be fairly accurate for me, but it's not going to be accurate for people who live in the US, whose actual genetic ancestry is a lot more sort of rich and diverse, and actually in the US, we have a lot more admixture. So like somebody whose grandparents came from Europe, but the other grandparent came from India, or that idea of sort of genetic admixture has to be counted when you actually want to sort of build meaningful models and insights from genomics. So the All of Us program is our opportunity to really create this population data set within the US and leverage it for this kind of research and make it available to all researchers. And so it's been doing that. Like I said, it's, hopefully it will continue to be funded to do that. But one of the things, early on, it really focused on the idea of short read whole genomes. But pretty quickly, the scientific community was asked, well, what would make this data set better? And we quickly said, well, you've got to add more types of data. Because we know what we're going to get to some extent from short read genomes, and we need lots of them, and it's really powerful. But really, to make it powerful, we need to add more things to it. And so the very first sort of new thing that was approved to be added to the cohort was long read genomes. And so across the academic groups, it was like their number one priority was like, we really got to go back into this population and start to show how much more we'll get if we actually add long read genomes to this data set. And so we've kicked off pilots. Kieran Garamella, Mike Talkowski, and others at the Broad and MGB have been sort of the academic leaders of that in our group. And with simple goals of apparently all the number one goal, but of enhancing our ability to actually find more structural variation. Because we know with short reads alone, we're missing about half of the structural variation. Figure out ways to actually construct resources to allow imputation into structural variant data sets from short reads and vice versa. And then also to really help catalog that new classes of variation that we're not going to get just from the short reads alone. And actually, once it's released, this pilot, sort of quote unquote pilot, of long reads within the All of Us cohort is about 4 times larger than any other population-based study that's been released to date for researcher access. And so we think this is going to be really, really powerful. The first phase of this data is already released in the researcher workbench for All of Us. It was about 1,000 participants sequenced at Hudson Alpha, lowish coverage, about 8x depth of a sort of largely, predominantly African-American cohort. So that data is already in the workbench for researchers. But the sort of the big bolus of the data is actually in phase 2, which represents about 14,000 participants. Participants were chosen in this, what we're calling the multi-omic cohort, not necessarily by phenotype, not for a particular disease state, but really to sample the cohort. So the value of the All of Us cohort is it represents that, again, that genetic ancestral diversity of the US population. And so, in the PC plot, you can kind of see the way we measure typically the spread of sort of ancestral genetic ancestry. And so the idea was to sample all parts of that space. And so it really kind of has a lot of admixture in between. It has trios. It has some other things that have been selected for other reasons. But so it's a really just representative data set. And then across different groups, Baylor and Broad, Hudson Alpha, Johns Hopkins, and UW, groups added different types of data. There's some amount of ONT, but the vast majority of this data set will be PacBio Revio data that we generated at the Broad. About just over 10 or 11,000 participants will have that data. That data is now, it had to go through we had to make novel methods to handle curation, variant calling of data of this scale, but it is now slated to be released in the workbench this summer. And so it will be available for researchers there, and there'll be a slew of other demonstration projects that come from just looking at this data. And I took a peek at it in terms of what are the phenotypes or the phe codes associated with these participants, and it represents, there's dozens and dozens and dozens of different diseases represented across this. 1,500 of the 10,000 participants have hypertension. There's all kinds of different phenotypes represented within this group. Likely we'll need to add more disease-specific groups to, this sort of in some ways forms the baseline against which you'll want to test now your ability to develop novel diagnostic modalities or insights from specific diseases. And actually, we have a follow-on study funded by another part of NIH to add to this data set another 2,000 long-read genomes from just participants within all of us who have diabetes. So that'll become like a disease-specific cohort. And then you sort of can compare that to your sort of baseline cohort. And the idea would be you'll have lots of these additional studies to then dig into the very rich data you can get from long reads and compare them to the All of Us data set. So really excited about that. We're also doing, in collaboration with PacBio, additional populations. So with Ben Neal's team and at the Broad Institute, we have a project to do Latin American genomes on long reads, and that data is actually being generated right now. So switching gears, the other very exciting project I want to talk about is a collaboration facilitated by PacBio between Broad and Boston Children's Hospital. Wendy Chung, Alan Beggs at Boston Children's Hospital, who collectively, of course, are really, really interested in childhood rare diseases. And the Boston Children's Hospital has, of course, the Manton Center for Orphan Disease Research, and they have a suite of researchers then who have sort of collected cases over the years of undiagnosed rare childhood diseases. And we really wanted to see, could we move the needle? And these cases have all been done with short reads. They've had exomes, they've had genomes, they've had lots of different testing modalities already. Could we eke out any additional information from adding long reads to these? And so this is really, Children's Hospital Rare Disease Collaborative, which is their sort of collaboration within their sort of researcher work groups to collect and have cohorts of patients with complex conditions who've gone through multi-year diagnostic odysseys. And so samples from children's came to Broad Clinical Labs, sequenced on Revio, and then returned to children's who did sort of the in-depth analysis with their in-house expertise. So it's great as well that sort of, we are not doing all the analysis for them. Children's Hospital has built its own ability to do, long-read analysis as well. And the idea, of course, you're getting all of these rich different variant types because you have the long reads. And so far we've got sort of 675 samples done or completed, delivered back. They represent, a range of different types of disease families that have been aggregated. I will say some of these are patients who've been collected years ago. So we had challenges with some of the samples are not great, and so we didn't get as high yields as we wanted. But this is reality. These are the samples they've got, and so it's what can we get from the data that's available? And so far, they're still working all the cases. We're still delivering cases to them, and they're still doing their analyses. But in different disease areas, we've had some really interesting findings. And in aggregate so far, at least 15% of the cases had new sort of findings that point to underlying disease pathologies. And that seems like a smallish number, but again, these are all already been tested by all the other modalities. So getting this actual additional insight is actually pretty encouraging. So I just did 2 use cases there of how we're leveraging long reads to supplement large-scale population data sets, starting hopefully to add disease areas into it, and then also powering this rare disease area. There's many other use cases that we don't have time to go into today, but I do want to say one of the things that people are really interested in, of course, from PacBio type data is you get this sort of methylation signals essentially for free. And so there are projects now that really want to use this methylation profiling as for biomarker discovery. And this really is to enable that translational space, which is exactly how we did it on the short read whole genome side. We have a whole bunch of research genomes, but then you want to start to enable translation, which means you want to enable clinical research. You want to enable those studies that maybe dual consent people into research, but also want to do return of results from them. You want to have a auditable quality system if people want to start to do clinical trials. And ultimately, then you might want to develop your own sort of complete sort of diagnostic report from the data as well. But we're starting the first step of that, and actually our validation, our technical analytical validation of the Revio is slated to be done by August. So all in all, it's a very exciting time in general. I think more important than ever is showing the utility of generating genomic data and how it translates into meaningful, actionable insights for patients, how it drives biomarker discovery and drug development, and how it sort of enables new discoveries. And then we have to take the pragmatic steps of like making it available to people who need it at the regulatory level they need it, and at the cost and turnaround times and everything else that they need it. With that, I got about 4 minutes left, so I'll wrap up and allow some time for questions. I just want to make sure I thank everybody who really contribute to this, the staff who run the instruments, our funding agencies, the collaborators who bring their science to us, and particularly the people who sign up for research, the participants, the patients and their families who allow us to do this type of testing for them. And if you ever need to connect to us at all about accessing our capacity to run Revios, please reach out to us, genomics at Broad Institute or broadclinicallabs.org. With that, I'll stop for questions. Thank you.